You know, you are quite literally made of virus. Right now, as you listen to this, about 8% of your DNA isn't actually human at all.
Yeah, it's just wild to think about.
Right. It's the leftover genetic ghost of these ancient viral infections that permanently spliced themselves into the human genome like millions of years ago. So welcome to today's deep dive. We have a seriously fascinating stack of sources for you today, a collection of foundational microbiology texts, some really old historical scientific papers, and modern biological essays. And they are all circling this one deceptively simple question. What is a
And it
virus?
really is a tough question.
It is. Our mission for you today is to explore what is arguably the ultimate biological identity crisis. We're going to look at why viruses blur the line between the living and the dead, and why science still, even today, struggles to classify them.
Well, to really frame this identity crisis, I think we have to establish the baseline standard first. The classic microbiological definition. It basically dictates that a virus is a submicroscopic infectious agent. It's made of genetic material, so that's either DNA or RNA, but never both. And it's wrapped in this protective protein shell called a capsid.
Right, a capsid.
Yeah. But the non-negotiable clause here, the absolute core of the biological paradox, is that a virus is an obligate intracellular parasite.
Okay, let's unpack this. Because obligate intracellular parasite sounds like a mouthful, but it means it operates with absolute biological minimalism. It has 0 ribosomes for protein synthesis, no mitochondria for generating ATP.
Nothing. It is none of the machinery for life.
Exactly. Outside of a cell, a virion is completely inert. It's biochemically closer to, like, a grain of quartz than a living bacterium. I always think of it like a USB drive sitting on a desk. That drive contains a highly specific, potentially destructive set of instructions, but it possesses absolutely no hardware to read or execute those instructions until it gets plugged into a computer, which would be the host system.
That's a great analogy. And taking that visualization a step further, that inertness on the desk is precisely why the standard scientific litmus tests for life produce such a fractured result. Because when biologists look for life, they generally demand 2 major criteria: independent metabolism and the capacity for Darwinian evolution.
And viruses fail that first test spectacularly, right?
Oh, completely. A virion resting on a doorknob is undergoing 0 metabolic chemistry. It isn't consuming energy. It isn't generating waste. But, and this is the kicker, they pass the Darwinian evolution test with flying colors. I mean, they mutate, they undergo natural selection, and they evolve to bypass host defenses.
Which makes total sense when you look at the mechanics, especially with RNA viruses, because RNA replication often lacks the proofreading enzymes that DNA polymerases have, right?
Yeah, they're incredibly sloppy copiers.
So they make constant copying errors. And most of those errors are fatal to the virus, but a few confer an advantage, which is exactly how something like influenza or, SARS-CoV-2 adapts so staggeringly fast. But I actually have to push back a bit on the binary nature of this.
Oh, how so?
Well, if an entity can mutate, compete, and evolve to survive this intense immunological arms race, isn't it just a little bit pedantic to declare it not alive purely because it doesn't digest its own food? I mean, it operates exactly like a living enemy.
You're hitting on a major fracture point in modern virology there. In fact, the biologist Luis Villarreal tackled this directly in a 2004 essay from our sources. He argued that viruses possess a genuine temporary living phase.
A temporary living phase.
Yeah. His argument focuses entirely on the intracellular state. When a virus actually enters a host cell, sheds its capsid, and begins actively transcribing its genome, that process is biochemically indistinguishable from the gene expression happening in any free living organism.
Okay. So the USB drive is alive the exact moment the computer starts reading the files.
According to Villarreal's framework, yes, exactly. But critics counter that this stretches the definition of life until it loses all meaning. Because if a virus is deemed alive simply because its genetic instructions are actively being transcribed by stolen machinery, does that mean any self-copying chemical network is alive? What about a rogue plasmid in a test tube?
Right, where do you draw the line?
Exactly. Ultimately, most working virologists just bypass the philosophical quagmire entirely. They accept the dual nature of the virus, often referring to them quite provocatively, I think, as organisms at the edge of life.
Organisms at the edge of life. That is an incredible phrase. But to really understand why we arrived at this super ambiguous definition, we have to look at the historical timeline of how we discovered them. Because for decades, the word virus which is literally just the Latin root for poison, was just a placeholder. It was a functional label for a phenomenon we couldn't even isolate or observe.
Right, the turning point for observation didn't come until the 1890s with the Chamberlain Pasteur filter. This was a piece of laboratory equipment made of unglazed porcelain. And the pores in this porcelain were so microscopic that it could physically strain out every known bacterium.
Which was the gold standard for sterilization back then.
Exactly. So in 1892, this Russian botanist, Dmitry Ivanovsky, was investigating tobacco mosaic disease. He crushed infected tobacco leaves, pushed the sap through this porcelain filter, and discovered the resulting clear liquid retained its full infectious capacity.
But he couldn't let go of the prevailing germ theory of his time, right? He just assumed it was a bacterial spore that was somehow tiny enough to slip through the porcelain matrix.
He did. The conceptual leap required to imagine something smaller than a bacterium was too vast at the time. It actually took the Dutch microbiologist Martinus Beijerinck in 1898 to replicate that exact experiment and push the interpretation into totally new territory.
Because Beijing noticed it didn't act like a solid at all.
Right. He noticed that the infectious agent diffused through solid agar gel just like a liquid chemical would. That is behavior that is completely impossible for solid bacterial cells. Furthermore, he proved it required actively dividing plant tissue to reproduce. You couldn't just grow it in a sterile nutrient broth like a bacteria.
It needed a host.
Yes, so he termed it a contagium vivum fluidum, a contagious living fluid.
A contagious living fluid. Just imagine the paradigm shift that required for a scientist back then. You've just convinced the entire medical establishment that diseases are caused by these physical, culturable, bacterial cells, and suddenly Beijering proves there's an invisible, unculturable, liquid pathogen out there.
It must have been terrifying.
Utterly terrifying. And it wasn't an isolated anomaly either. The sources note that foot and mouth disease in cattle was identified as a filterable agent that exact same year. And soon after, Walter Reed demonstrated the same thing for human yellow fever. This invisible fluid was everywhere.
And its true physical structure remained entirely theoretical until the 1930s. What finally shattered the whole living fluid concept was the American biochemist Wendell Stanley in 1935. He actually managed to isolate the tobacco mosaic virus and achieve something previously thought impossible for a pathogen. He crystallized it.
Wait, hold on. He crystallized it like a mineral phase change, like salt. How does a biological entity survive being precipitated into a crystalline structure without all its proteins completely denaturing?
Well, what's fascinating here is that the extreme structural symmetry of a viral capsid, we're talking millions of identical protein subunits locked together in a highly ordered geometric lattice, it lends itself perfectly to crystallization. Stanley produced these pure, needle-like crystals that behave like a completely inert chemical compound. You could just store them in a glass jar on a shelf indefinitely.
Wow.
And this is the crazy part, the moment you dissolve those crystals back into an aqueous solution and apply them to a host organism, they initiated a biological infection.
See, that is the exact moment the philosophical debate becomes mathematically unavoidable. You have an entity sitting on a shelf, physically indistinguishable from a jar of crystalline salt, but you add a biological host and it instantly executes a targeted genetic takeover. Which forces us to ask about the mechanics here. If viruses are just geometric protein crystals, how do they actually initiate that cellular hijacking?
It relies entirely on a process called host tropism, and that is driven by molecular surface tension and receptor affinity.
Right. I've seen it described as a lock and key. But honestly, looking at the biochemistry in the sources, it behaves much more like a biochemical barcode scanner.
A barcode scanner. I like that.
Yeah, because a virion just drifts through the extracellular space randomly until the glycoproteins on its outer surface brush against the host cell. It doesn't just bump into any cell and infect it. It requires a highly specific docking sequence. HIV, for instance, has a surface glycoprotein that specifically scans for and binds to the CD4 receptor on human T cells.
It's highly, highly targeted.
Exactly. If the biochemical barcode matches, the viral capsid fuses with the membrane, injects its genetic payload, and overwrites the host cell's native programming. The host cell just stops synthesizing its own necessary proteins and becomes a localized virus factory.
And because that genetic payload is literally the only functional asset the virus brings to the table, virologists realized pretty quickly they couldn't organize viruses based on morphology or metabolism. So in 1971, David Baltimore revolutionized virology by proposing a classification system based entirely on genetic strategy. The Baltimore classification system organizes all viruses by how they synthesize messenger RNA.
Because messenger RNA is the universal language of the host's ribosomes. No matter what a virus is made of, whether it's DNA or RNA, it must eventually produce mRNA to trick the host into manufacturing viral proteins.
Precisely. Every virus is essentially taking a different path to the same destination. Double-stranded DNA viruses, like the herpes viruses, they enter the host nucleus and simply co-opt the host's standard transcription machinery, much like our own genes do.
Right.
Single-stranded RNA viruses, on the other hand, like poliovirus or SARS-CoV-2, they can bypass the nucleus entirely. Their genome can often be read directly by host ribosomes right there in the cytoplasm. But the most radical pathway belongs to the retroviruses.
Like HIV.
Yes. Retroviruses carry a special enzyme called reverse transcriptase. When they inject their single-stranded RNA into a cell, they deploy this enzyme to transcribe their RNA backward into double-stranded DNA.
Which breaks all the normal rules of biology.
It does. They then utilize another enzyme, integrase, to splice that newly synthesized viral DNA directly into the host's chromosomal genome. They literally rewrite the biological blueprint of the host cell.
That's just wild. But, I want to talk about the physical packaging required for this payload delivery because there's a brilliant evolutionary divergence here. If a virus is essentially just minimalist genetic code encased in a protein capsid, why do some viruses require an extra external lipid envelope like influenza or HIV, while naked viruses, like the rhinovirus that causes the common cold, operate just fine without one?
That divergence in packaging strategies really highlights their role as biological scavengers. See, enveloped viruses don't possess the lipid synthesis pathways to manufacture that outer layer themselves. Instead, as the newly assembled virions prepare to exit the host cell, they migrate to the cell membrane. They push outward, pinching off a section of the host's own lipid bilayer, and they wrap themselves in it like a stolen cloak.
They steal the curtains on the way out. And they do it to disguise themselves from the host's immune system, right? Because to a patrolling white blood cell, that lipid envelope looks remarkably like a native cell membrane.
It's perfect camouflage.
Naked viruses, on the other hand, they don't bother with stealth at all. They just trigger cellular lysis, literally bursting the host cell open and flooding the surrounding tissue.
Which perfectly encapsulates the elegant yet terrifying simplicity of viral mechanics. But, just as the scientific community settled into this comfortable consensus of viruses as minimalist, submicroscopic genetic scavengers, the biological world revealed an entirely new tier of complexity.
Oh man, here's where it gets really interesting. Because if viruses are defined by their reliance on stolen machinery, there should be a hard physical and genetic limit on how large they can get. But in 1992, researchers investigating a pneumonia outbreak in Bradford, England, isolated a particle from a cooling tower that defied every established rule of viral geometry.
Yeah, the Bradford particle. It was initially an epidemiological dead end. It measured roughly 750 nanometers in diameter. It was so massive that it absorbed gram stain and was actually visible under standard optical microscopes.
Which viruses aren't supposed to do.
Right, so the researchers logically assumed it was just a particularly stubborn bacterium. They stored it in a freezer and largely forgot about it for over a decade.
Because it completely violated their mental model. A virus physically cannot be that large, so they just assumed their diagnostic tools were picking up a bacterial anomaly.
That cognitive bias held until 2003. When French researchers Bernard Lascola and Didier Raoult finally subjected the particle to electron microscopy and genomic sequencing. And the results were just paradigm-shattering. It was definitively a virus. They named it mimivirus, standing for mimicking micro. But its sheer physical size was only the beginning.
Right, the genetics were the real shock.
Exactly. The mimivirus genome contained over 1,000 distinct genes. For context, many free-living parasitic bacteria operate on fewer genes than that. And then a decade later, the discovery of Pandora virus pushed the boundary even further, boasting a genome exceeding 2 million base pairs.
Over 2 million base pairs. And the most staggering detail from the sources is what those base pairs actually encode. Because these giant viruses carry genes that code for components of the protein translation apparatus, aminoacyl tRNA synthetases.
Right, which breaks all the rules.
Yes. The absolute foundational rule of virology was that viruses lack translation machinery, making them obligate parasites. Discovering translation genes inside a viral genome fundamentally breaks that definition.
It forces a total reevaluation of evolutionary biology. If we connect this to the bigger picture, the standard phylogenetic tree of life we've utilized for decades, divided into bacteria, archaea, and eukarya, suddenly seems inadequate. The sheer complexity and unique genetic repertoire of giant viruses have led several prominent evolutionary biologists to argue that these entities actually represent a distinct fourth domain of life that evolved parallel to cellular organisms.
And as if a fourth domain of giant viruses wasn't enough to process, the very concept of parasitism gets turned inward with the discovery of the Sputnik virophage.
Well, Sputnik is fascinating. Discovered in 2008 by the same Parisian research team, the Sputnik virophage represents a bizarre biological nesting doll. It is a virus that specifically infects another virus.
A parasite hijacking a parasite. I mean, how does that even biochemically function?
Well, it requires a highly specific environment. When the giant mimivirus infects an amoeba, it constructs a massive, localized viral factory within the amoeba's cytoplasm to coordinate its immense genome replication. The Sputnik virophage cannot replicate in an amoeba alone. It has to wait until the mimivirus establishes its factory.
And then it strikes.
Exactly. At that point, Sputnik infiltrates the factory and hijacks the giant virus's transcription machinery. Sputnik replicates at the expense of the mimivirus, physically stunting its growth and reducing its infectious yield, and essentially makes the giant virus sick.
Which naturally forces us into the deepest question the sources tackle today. Where did this incredibly diverse, nested hierarchy of genetic code actually come from? If viruses range from these minimal RNA strands to giant DNA behemoths that have their own parasites, they clearly didn't all evolve from a single common ancestor.
No, they didn't. The scientific consensus overwhelmingly supports a polyphyletic origin, meaning different classes of viruses emerged through entirely different evolutionary pathways. Virologists generally categorize these pathways into three competing hypotheses. The first is the regressive hypothesis.
Which argues what, exactly?
It argues that viruses are the degenerate descendants of once free-living, complex cellular organisms. Over millions of years of parasitism, these cells steadily lost the genes required for independent metabolism, stripping down to bare biological essentials.
Which the giant mimivirus heavily supports, right? Because it looks exactly like a cellular organism caught halfway through the process of evolutionary degradation. But what about the smaller viruses? They don't look like degraded cells at all.
Right, and that leads to the second framework, the progressive or escape hypothesis. This model suggests that viruses originated from within the host cells themselves. We know genomes are incredibly dynamic elements like transposons or jumping genes, frequently excise themselves and move around the chromosomes. The escape hypothesis posits that long ago, some of these mobile genetic elements accidentally acquired structural protein coding, broke free of the cell membrane, and basically achieved autonomy as infectious agents.
So, rogue cellular code that broke out of the nucleus, that makes sense. And the third hypothesis.
The third is the virus-first hypothesis, which flips the timeline completely. Drawing on the RNA world hypothesis, this model argues that self-replicating RNA molecules predated the evolution of cellular life entirely. In this primordial environment, viroid-like entities were the dominant self-replicating structures. So in this view, viruses aren't a byproduct of cellular evolution. They are the ancient bridge from prebiotic chemistry that eventually gave rise to the first true cells.
Understanding that deep evolutionary history completely changes how you view a simple viral infection. And it also highlights a massive public misconception regarding how we treat them, specifically regarding antibiotics.
Oh, this is a huge one.
Yeah. The misuse of antibiotics against viral infections like the common cold or COVID-19 isn't just ineffective. It fundamentally misunderstands biological mechanics. Because antibiotics are highly specialized chemical weapons designed to target and dismantle independent cellular machinery. They destroy bacterial cell walls, or they inhibit bacterial ribosomes.
Exactly. So applying an antibiotic to a viral infection is biochemically useless. It is akin to pouring herbicide on a rock. You are attacking metabolic processes that simply do not exist within a viral capsid.
Pouring herbicide on a rock, that is the perfect way to put it. And that distinction extends to our vocabulary of sanitation, too. You don't kill a virus when you bleach a countertop. You inactivate it. The chemical agent denatures the structural proteins of the capsid, destroying the geometrical precision of that biochemical barcode scanner we talked about earlier. Once the scanner is warped, the virion loses its infectious capacity, but it was never metabolically alive to begin with.
Right. And shifting our perspective from eradication to understanding reveals perhaps the most profound revelation in our sources today. The realization that viruses are not merely external pathogens. They are foundational architects of mammalian biology.
Okay, this is where we have to talk about syncytin, because we view viruses purely as agents of disease. But syncytin proves they are inextricably woven into our survival. Millions of years ago, an ancient retrovirus operating with the exact same reverse transcriptase mechanics as modern HIV infected the germline cells of a primitive mammalian ancestor. It permanently inserted its viral envelope gene into the host's DNA. But instead of causing a lethal infection, natural selection captured and domesticated that viral code.
And the biochemical application of that domesticated gene is astounding. The viral envelope protein originally functioned to fuse the viral membrane with the host cell. Well, the mammalian body repurposed that exact fusion mechanism to create the syncytiotrophoblast.
The syncytiotrophoblast, what is that for the listener?
It's a continuous fused layer of cells in the placenta. This barrier is entirely reliant on that ancient viral protein. It mediates nutrient exchange while simultaneously cloaking the developing fetus from the mother's immune system, utilizing the very same immune evasion tactics the retrovirus originally used.
That is just incredible. Without that ancient retroviral infection, the evolutionary pathway of placental mammals, which includes us humans, would not exist. We would still be laying eggs. And the irony is, modern medicine is finally learning to consciously direct that exact same hijacking logic. We are turning viral mechanics into therapeutic tools today.
We really are. The mRNA technology utilized in the COVID-19 vaccines is a direct application of viral strategy. By encasing synthesized mRNA in a lipid nanoparticle, we mimic the viral entry mechanism to deliver a localized instruction set, basically forcing host cells to manufacture harmless spike proteins for immune training.
Right, we're acting like the virus.
Exactly. Furthermore, in modern gene therapies like CAR T-cell treatments for oncology, we utilize heavily modified lentiviruses. We strip them of their pathogenic payload and replace it with therapeutic genetic edits, utilizing their natural nuclear integration tools to rewrite defective cellular code.
We're even dusting off bacteriophages, the viruses that exclusively prey on bacteria, as highly targeted biological weapons against antibiotic-resistant superbugs.
It represents a total paradigm shift. We have evolved from fearing Bacharach's invisible living liquid to actively domesticating viral mechanics to engineer human immunity.
So, to summarize the immense territory we've covered for you today in this deep dive, we started with the porcelain filters of the 1890s, straining out this unculturable biological mystery. We watched Wendell Stanley cross the boundary of life and chemistry by crystallizing a pathogen. We mapped the genetic hijacking strategies delineated by the Baltimore classification, from DNA co-opting to the reverse transcription of retroviruses. And we expanded the boundaries of biology with the discovery of giant viruses carrying translation genes and virophages parasitizing other viruses.
And underlying all of this incredible biological diversity is one inescapable truth. Regardless of whether we definitively classify them as living organisms or complex biochemical machines, viruses are undeniably the most potent, ubiquitous drivers of evolutionary change on Earth. They are the ultimate biological fluid, constantly forcing us to redefine the parameters of life itself.
Which leaves me with a final thought for you to ponder. We talked about how an ancient retroviral infection millions of years ago provided the crucial genetic hardware for the mammalian placenta. It fundamentally altered the trajectory of our species. It makes you wonder, as our genomes interact with the viral pandemics of the modern era, what new genetic code is actively being written into our DNA right now? In a million years, what entirely new human trait, what new biological capability will exist solely because of a virus we are fighting today? Thank you for joining us on this deep dive into the microscopic world. Keep questioning, keep exploring the sources, and we'll see you next time.